Okay, we'll see if we've got the technical issues worked out and we will finish up this section. Hopefully we'll get through section 6.3 and talk about protein folding and domains. Okay, so besides X-ray crystallography, you can also use NMR, nuclear magnetic resonance spectroscopy. I'm not going to spend a whole lot of time on this because it should be familiar to you from organic chemistry. Essentially, we can probe the nucleus of an atom, specifically those of hydrogen and carbon-13 nucleus, and they actually have a property called spin. And if you put them in a strong magnetic field, you'll separate the nuclei into two different levels, the alpha and the beta state. They're roughly equal in those two states. You get about 1 in a million more. So about 1,000,001 in the, 1,000,001 in the alpha state and 1,000,000 in the beta state. That just lets you know that the energy difference between those is roughly equal, but you get just slight more in the alpha state, about, like I said, 1 in a million. And if you hit it with the right amount of energy, since it's quantized, you can actually get those nuclei to flip. So again, that's what we talked about in organic chemistry, but in protein chemistry, we have some additional problems. And you can see that in this NMR spectra to the right. You can see that it's just really, really information dense. I won't say noisy because those peaks each correspond to a hydrogen, but there's just a lot of information in there. There's just too many protons to really resolve. Where you see the one off to the left there, that's ethanol. Those are the type of NMR that we're used to seeing, very resolved peaks, lots of, well, characteristic splitting that you can then identify the structure from. So just too much information for a protein. So what they do is referred to as a 2D NMR. So you're looking at 2 dimensions. And in those 2 dimensions, there's several different things that you can look at. One of the most powerful is what's referred to as the nuclear Overhauser effect. And so it identifies interacting protons that may not be adjacent to each other. So just like we talked about in NMR, the protons on one molecule can interact or influence the neighboring protons because they behave like tiny magnets. And so that's also true through space there. And so if you look at protons 2 and 5, you can see that they actually are close enough. If they're within a certain distance, they can actually interact and cause a peak or influence that. So what they refer to as an off-diagonal peak. Now these on the diagonals here, these are the peaks corresponding to the protons 1 through 5. But if you get this through space interaction, you'll see an off-diagonal peak that corresponds to the interaction between proton 5 and proton 2, as well as proton 2 and proton 5. And so that lets you know that those 2 protons are in close proximity to each other. And so that can be very powerful because instead of getting 5 different signals here, you essentially get a mirror image of 1, and that lets you know who's close to each other. So here is a picture of what they refer to as a noisy spectrum, nuclear Overhouse effect spectrum. And again, along here is what you'd see for a normal one-dimensional NMR. But then these off axes here, so these are the peaks that correspond to the corresponds to the proton signal there and the proton signal there. So there's some type of interaction. And you can see that there's a lot less of those off axis than there are on the axis here. It does get a little bit congested up in this region, but if you use a bigger NMR, these spots become more resolved. And what I mean by a bigger NMR is just more, a larger magnet, higher operating frequency. So if we have our protein over here from N terminus to C terminus, we're just looking at a few signals for A, B, C, and D. And so you can look at those signals. So here's the signal for D, here's the signal for A, and so here's the interaction between A and D. Here's the signal for C, there's the signal for D again, and so there's an interaction. Here is signal C, and it looks like it's interacting with signal B. And so from there, what you can do is you start to figure out that A and D have some close association. What did we say? C and D have some close interaction there. And then C and A, what's this one right here? C and B. Oh yeah, sorry, C and B have some interaction there. So you start to make these models of who can be close to each other. And again, the full details of this beyond the scope, but what you end up with is a bunch of different models of where that backbone could be, because the intensity of those spots will change. And the other thing is you're determining this in solution. And so NMRs can breathe and the backbone can flex. But you take those NMR spectra, and you can get many of them. And then what you do is you come up with a model here. And so you can see the backbone in white here. And you can see in some regions that that backbone looks like it's moving. And so how you know it's moving is those off-diagonal peaks would increase or decrease in intensity. And then there's some peaks that are going to be very constant in their intensity. And that tells you that that region is fairly defined. And so you don't get a single structure here. You actually get a bunch of overlapping structures, a model of showing the flexibility of the backbone. But if you take this model and you overlay it with a 3D X-ray crystal structure, they tend to match fairly closely with the positions of the side chains in the backbone. So each of these have their own uses. So here's some compare and contrast. So using NMR, it's difficult to solve large structures. So typically the protein needs to be 100 kilodaltons or less. And the reason for that is you just have too many signals. But they're finding ways around that. So they're pushing the upper limit to maybe even 1,000 kilodaltons, which is actually 1,000,000 daltons. So they're starting to be able to look at larger proteins. So it's amazing what they can actually do with NMR now to actually get this information. Proteins need to be labeled with NMR active nuclei. So those are hydrogens and carbon-13, but remember carbon-13 only occurs 1 out of every 100 atoms. And you can use nitrogen, but you have to use a isotope of nitrogen that's NMR active. So often you have to grow up the bacteria or make the proteins with more active NMR nuclei to get enough. So that is a drawback. But NMR can be used for proteins that don't crystallize well. And NMR can also show protein movements that may be lost when the protein's actually stuck in a crystal. Some disadvantages to crystallography is it may be a pain to crystallize proteins, and some don't give high resolution proteins. So you may get a crystal, but it won't diffract. Crystal structures capture single states, but crystallography is much more possible for large proteins and complexes. But in the end, high resolution crystallography shows conformations much more exactly than NMR does. So that is one advantage for crystallography, but they can be used together. Now there is a new technique that is coming online, and it's been around for a while, but it's getting more and more powerful every month. And that method is cryo-electron microscopy. So I remember a long time ago thinking about, well, why can't you just shoot electrons? We can do scanning, tunneling, microscopy, and electron transmission or microscopy and look at really, really small things. So I remember thinking, why can't you just shoot an electron beam and get the shape? Well, there are a couple reasons at the time that you couldn't do it, but you can do it now. And one of the reasons is you have to place these molecules into a vacuum. And when you place things into the vacuum, the solvent tends to evaporate off or sublimate off. So what they do here is they actually freeze these samples with the water around them in what they call a flash freeze. So the ice or the water actually doesn't crystallize into an ice form. It actually is more glassy, amorphous. If the water starts to crystallize, that starts to cause deformations in the structure. So what you do is you flash freeze these, and then you shoot an electron beam at them. And essentially you can get a picture, but that picture is only going to be 2 dimensions. I've got another picture on the next slide here that shows a protein complex. And when you take a picture, again, it's only going to be 2 dimensions. And what you need to do is actually take pictures of this protein at several different angles. So if you deposit this protein on the surface, you're going to have some that have the top view and some that have a side view and some that have a rotated side view and some that have a bottom view. And when you hit this with the electrons, essentially you can get a picture of the molecule, and they're actually getting where they can get really high resolution of these. And so this is a really good experiment if you have a really large assembly of proteins. And then they do a bunch of calculations to figure out this shape and turn it into that. And you may say, that's kind of a stretch there. I believe in a lot of cases they know the sequence of the protein and they have some other information. And so they use this in conjunction with either NMR or X-ray crystallography to get these structures. Otherwise, this right here, you can kind of see the general shape there, but it seems like maybe there should be more protein here. Again, the scope of how they actually do this is a little bit beyond us, but this cryo-electron microscopy is becoming very, very useful, and they're starting to get really high resolution. And so the advantage of this is you don't need to prepare crystals, and you just essentially deposit the protein and flash freeze it, and then you capture it in a bunch of different states. Okay, so that's another way that we can get to the 3-dimensional structure of proteins. So when they go and look at the distribution of where protein or where amino acids appear in these protein structures, you start to get some trends. So non-polar residues mostly occur in the interiors of proteins. So you don't have these greasy groups really on the surface of proteins. They tend to tuck themselves away from contact with water. Now, charged polar residues are usually on the surface, and that's because if I put a charged residue right in a anhydrous or greasy interior, that's a really high energy position. Now, we can bury these charged polar residues on the interior of a protein if they happen to be paired with an opposite charged amino acid. I believe we'll talk about that a little bit later. And then you've got these uncharged polar residues, and they occur in both places, on the surface and on the interior. If they're on the interior, then they always tend to be hydrogen bound with something else and just not dangling out in a greasy or among the hydrophobic or nonpolar residues. So these tend to hydrogen bond with other residues or the backbone. Okay, so let's look at some protein structures and look at where the positions of their side chains are. And what we can see here is an alpha helix from myoglobin. And so the backbone is in gray, the non-polar side chains are in gold, and then the polar side chains are in purple. And you can see roughly that they tend to be found on one side or the other. So in this alpha helix here, you can kind of see that the purple are all kind of pointing out in one direction, and then the gold, the non-polar, are all on one side. And then these would be found towards the interior of the protein, or maybe these are buried in a lipid. And then these are what are referred to as solvent exposed. You can see in a beta sheet here, you can see on one side, and remember in a beta sheet, it alternates the amino acids, kind of one's on top, one's on bottom. And you see, again, generally that the nonpolar are on one side and the polar are on the other side. You can see some exceptions to those. Okay? These aren't hard and fast rules, but in general, it makes more sense to put the hydrophobic residues with the hydrophobic and the polar with the polar, or the polar exposed to the solvent. And that's just the same picture. So here is another picture. This is an artist rendition. And so this is the nonpolar and polar side chains in horse heart cytochrome, just showing that the polar that are green, they tend to be kind of more dangling towards the outside of the protein. And then the red orange-ish, they tend to be buried. And these are just the side chains, these colors, they tend to be more buried inside. But you can see this valine that's kind of pointing out towards the solvent there. So it's not always true, again, but generally you want to bury those hydrophobic residues in the interior and have the polar residues pointing out. And again, sometimes you see a polar residue right here. I believe this is histidine, and that plays a very important role that we're going to see later on. And so sometimes you bury these polar residues in the interior, but that's because they have an important job to do. And something else when we talk about three-dimensional structure that we need to mention that's very, very important, and you actually can see this in crystallography and maybe even in NMR, depending how you do it, but that is ordered water. So protein interiors are generally closely packed, and they exclude what we call bulk water, meaning you just don't have a bunch of disorganized water in there, but sometimes you can capture a water within the interior protein, and you can see it in the three-dimensional structure. And these are functional waters. They're either proton carriers, they're involved in hydrogen bonding, or they're kind of bridging the two strands to allow hydrogen bonding between two strands or two amino acids that are maybe distant, or they're coordinating a metal compound inside. So you can find waters that actually are very ordered inside of proteins. And again, those are going to be waters that are involved in hydrogen bonds with either the backbone or side chain residues. And they often have a purpose, either in the reaction or helping the protein adopt a given conformation. Now, you're not really going to find those waters buried in hydrophobic regions, but like I said, you can see the polar amino acid residues in the interior. They just tend to be hydrogen bound. And so it may be a water that is bridging those amino acids. So anyway, when we talk about enzymes, we'll see that those water molecules are important. Okay, all that discussion about how do we get the three-dimensional structure of proteins leads to this point of how do we know the tertiary structure. So the tertiary structure is once we take the primary structure, which is a sequence of amino acids, and we get a secondary structure, which are these beta sheets and alpha helices and other, locally organized regions of the polypeptide. Well, those locally organized regions can actually organize into higher organized overall polypeptides. So in tertiary structure, we look at all these locally organized regions and how they arrange themselves in an overall peptide. And so a lot of times these combinations, you see these regular arrangements. So here we've got a beta strand with an alpha helice followed by a beta strand. Sometimes you see the beta, this would be the anti-parallel beta sheet, but this is what you could also refer to as a beta hairpin. You may see alpha helices that cross each other, and you'll notice if you looked at the interior or the contact surface between those, it actually had hydrophobic residues there. Here's kind of two beta strands running, and then they've got this region here that can actually fold into what's referred to as this Greek key motif. So you kind of got anti-parallel beta strands, but you've got these close linkages, and then you've got a really long linkage. And the reason they call it a Greek key is it matches this Greek architectural design feature. So these are what we would call kind of super secondary structures. So they're still kind of secondary structures, but this is what we see when we start looking at the overall arrangement of the tertiary structures. So tertiary structures aren't just going to be alpha helices and beta sheets. Sometimes you're going to see some of these arrangements here. And so here is some examples of proteins, and we're going to see that you can classify them based on their alpha helical and beta sheet content. So off to the left, we would just refer to this as an alpha helical protein because it really just contains alpha helices, and there are some turns in there. And then in the middle, you actually have just a beta sheet protein. It's kind of coiled up there, but really just have beta sheets with some coils, and then you have some alpha beta proteins that have both. You have alpha helices and beta sheets in there. So we're trying to classify the tertiary structure based on these secondary structures in the proteins. So here's some more for tertiary structures. So again, we got beta sheets, that's a secondary structure, but if that beta sheet folds up into a barrel, then we refer to that as a beta barrel. And there's a couple different types that you can make that beta barrel. So you can see the one off to the left, beta barrel, and that's very similar to the green fluorescent protein, which also is a beta barrel. Then you've got this beta hairpin barrel, and this one's kind of like you rolled up a beta barrel. So if you looked at it kind of on the end, it would look like a spiral coming out instead of the one on the left would, if we looked at it on the end, would be a circle. And this one would kind of have a spiral like a jelly roll or a cinnamon roll or a Swiss roll. So this is what's referred to as the jelly roll motif. And then you have this Tim barrel that has some beta sheets, but also some alpha helices around there. So these are just some of the categories or topologies that you get for tertiary structures. And so often what we'll say are these, we've got these protein motifs. Now we've talked about protein domains before, but these motifs are actually these small repeating or common features that we'll see. And so one of them that you'll commonly see is an alpha helix. And then what that alpha helix does is that it has a turn, and then you'll just get into another alpha helix. So this is what we would refer to as a helix turn helix. So really they're just descriptive terms that you use to describe what you see. So again, here we have an alpha helix, and we've got a turn and then an alpha helix. So you got a helix turn helix. So it's just kind of a descriptor of what's going on there. So these motifs, what we find is they often have common features. So this helix turn helix motif is common to proteins that bind DNA. Now, when we talk again about motifs, they're kind of these common features that if you could look at. So this almost looks like a jelly roll. I'd have to look at it kind of on the end, but that may be a jelly roll motif. But this is a single polypeptide. And so what you have are these different domains that are connected by these flexible regions. And each one of these domains likely has a purpose or a compound that it selectively binds. So even though it is a single polypeptide, it has several different domains that are going to do different things. So again, it could either bind something, or it could catalyze a reaction, or it could be recognized by another protein. And so another protein actually binds here and then reaches over and does catalysis on a group that is fixed here. So here we would say that it has 4 domains. So in the past, when we've talked about domains being conserved, this is what we refer to, are these domains, these little functional pieces there. And so proteins will often swap these domains. And again, these domains often have a given purpose. So if whatever this piece here, if I needed that function and I included it in another protein, that could be evolutionary advantageous because now I've got that functional piece connected with another protein. Okay, so like here's an example here. We've got 2 domains here, and the bottom blue domain, I believe, is referred to as the Rossmann fold. And what it is it's a beta, alpha, beta units. And this Rossmann fold will actually bind the dinucleotide NADH. And so this Rossmann fold is actually a common domain that you see when you're binding NAD. So they often have specific functions and characteristics. Okay. So I'll let you read this on your own if you wanted to read it, but just really a description between the difference when we talk about motifs and domains. So motifs, again, are just kind of structural features. Domains are actually functional. And so, like it says, probably the most important thing. Theoretically here, you could separate the domains from each other. And then if I had a DNA binding domain, it will still bind DNA. And if I had a catalytic domain that was connected, it would still be catalytic. So domains are these functional regions where motifs are similar 3-dimensional structures conserved among different proteins. And they can be somewhat interchangeably used. But the structure of domains are more conserved than sequences. So there's as many as 1,400 protein domain families. So again, these are kind of the functional parts of the protein. But about 200 of these folding patterns account for half of all known protein structures. So these folding patterns that we see are very conserved. But that doesn't mean that the amino acid sequence is conserved. It just means that we get these typical folding patterns. So the domain is the apparent fundamental unit of protein evolution. And so the mutations are selecting for or against stabilizing this domain, or activating or inactivating those domains. So that's kind of the key that you want to take, is a domain is kind of a functional unit of the polypeptide. And apparently the most common protein domains are evolutionary sinks, meaning that we tend to get to that point and find out that they form stable folding patterns. They tend to tolerate deletions, substitutions, and mutations and still remain functional. And they also can support essential biological functions. So these domains end up being common, and they tend to perform common functions, but they do tolerate deletions and substitutions and mutations, and have similar but not identical folding patterns. So in fact here, we've got cytochrome C proteins and the related prokaryotic cytochrome C type proteins. So they're widely divergent sequences, meaning that if you looked at their primary sequences, you're not going to see a lot of connectivity or similarity between them, meaning you're going to have a wide variety of different amino acids at different places there. And if you look at these overall, just kind of squint really hard, you'll see that their shapes are somewhat different, if not very different. But these all have come up with a way to fold to get a heme group in the interior. So the interior folding is very similar, even though the outside may be very different. So the key is there's probably lots of different ways to get these amino acids in this orientation here. And so many different species have found ways to do that. And then what happens out here are you get mutations, and we can just accept those mutations and the protein remains functional. So really the biggest difference that we see here are the surface loops, and they don't affect the fold overall too much. If this is the functional portion, this is the part that needs to be organized. The rest of this here could be a tag for another protein, or as long as it gets the group further down in the right space, it will serve its purpose. Okay, I'm not going to spend a lot of time on bioinformatics. If we were in class, we would have some exercises on this, but in short, we're short of time. So there's lots of different programs out there to search for these structures. So we've got these structural databases. Probably the best known one is the Protein Data Bank, the PDB. We do have one for nucleic acids. We've got some molecular modeling databases, and then some NMR structure. But like I said, probably the most common one is a Protein Data Bank. Then we've got ways to view these structures. And so they give you a whole list here. And if you were in Howard's class, he would have you navigate to PyMol, which is his favorite. The one I'm most familiar with and have played with the most is known as VMD. I believe it's Visualization Molecular Dynamics program. And there's lots of these out there. Lots of molecular graphic programs allow you to do different things. And so you can actually open up the crystal structures and probe. And like I said, if we had more time and just weren't worried about as much about content, we'd take some time. These programs tend to have a little bit of a steep learning curve to get familiar with them. But if you go to the PDB, you can actually just type in and search for proteins there, and they have a little viewer that you can actually go and look at protein structures there. And then they have classification algorithms, so ways to compare and contrast proteins. And one of the reasons that you want to do that is for evolutionary comparisons. So if we wanted to figure out how closely related proteins are, we could use some of these resources down at the bottom. So as I mentioned, we're not going to spend too much on that section on bioinformatics there. So in fact, we will stop it right there for checkpoint 6.2. But we want to finish up the section on protein structure. So we're going to talk about quaternary structure and symmetry. So really out of section 6.3, we just want to talk about how some proteins contain multiple subunits, usually arranged symmetrically. That is the key concept. So many proteins are composed of multiple polypeptide chains, and those are referred to as subunits. And if you have more than one subunit associated together, that's referred to as quaternary structure. How do multiple polypeptides interact with one another? And so quaternary structure can be as simple as 2 identical polypeptide chains, or as complex as a dozen of different polypeptides change. So this one here is what we refer to as a homodimer, meaning that both the groups are the same, but they associate together in a dimer to form the functional protein. So here's an example. In A, we have a heterotetramer. So there are 4 subunits there. Now, the term hetero means that they're going to be different. And so in this case here, there are 2 identical subunits referred to as alpha, and 2 identical subunits referred to as beta. And so this is actually the structure of human hemoglobin that we have a whole chapter on. And so this is a heteroheptamer made up of 2 identical beta subunits and 2 identical alpha subunits. And it also has what's referred to as a prosthetic group or a heme group, and there's 4 of those. Right. Here is an amazing one. This is 60 copies of 40 subunits. So this is the coat of the human rhinovirus that causes the common cold. It comprises 60 copies of each of the 4 subunits. And so shown here are just 3 of the most prominent subunits as different colors. So I don't know if the other subunit is hidden in there, but anyway, I just thought this was an impressive structure showing you the complexity that can arise in quaternary structure, having many subunits associated together. So why do proteins have multiple subunits? This is important to know. So having many subunits instead of a huge polypeptide makes it easy to replace a defective part of the protein. So instead of having one tire, if you had 4 tires and one goes flat, it's going to be easier to replace that tire. Plus, it's easier to ride on 4 tires than on a unicycle. Because the unicycle, you have to pedal, and a car, you just have to push the gas pedal. Increasing the size of protein allows more control over and stability of its active sites. So this is probably why we also have proteins instead of smaller catalysts. What it means by that, instead of just a small molecule, if we have a large protein, you can actually start to tweak that active site very finely. But again, if we get too large of a protein, we run into problems. Maybe it's more difficult to make, and we'll talk about it in the next section. Folding is really a big issue. And so having multi-subunit proteins allow us to assemble smaller parts into a much larger functional unit. So multi-subunit proteins can allow for structural regulation and activity. And again, when we talk about hemoglobin and myoglobin, you'll start to see how having multiple subunits is actually beneficial. And then finally, multiple subunits with different functions have already evolved. So such as, like I mentioned earlier, the Rossmann fold that binds DNA. And so you can repurpose that. So what you can do is form a quaternary structure with these different functional subunits and bring those different functional subunits together to make a overall functional protein. Now, they talk a little bit about symmetry there. But in the end, there's very little that we need to know. So they give you a lot of these diagrams and they look fairly complex, but in reality, they're just talking about the different types of symmetry that you can have. And so you have this rotational symmetry, and that's what you see in this line A right here. And what that means is you can have many of these subunits rotated or oriented around an axis here. And kind of how many you have is what it's referred to. It's a little bit more involved than that. This is, a little bit beyond this scope. This is what's known as, oh, shoot. Oh, brain fart. Maybe this will be the bonus. Let's see if I can come up with it. Oh, it's not coming to me. We'll see if we can. Oh, it's on the tip of my tongue. Anyway, there is a field where you talk about symmetry. There we go. So this will be our bonus. The field or the topic when you're talking about symmetry. What is that called? I can't believe I can't remember that. Anyway, but there is kind of a general classification for talking about symmetry. Anyway, if you probably looked up C2, C3, C5 symmetry, C7, D2, D3, D4, and symmetry operations, you would be able to answer that question that I can't right now. Anyway, across the top, you see these that have rotational symmetry. So it's really the number of subunits that you have and the way that you can rotate or flip them to end up back at the identical point. So this one's C2 symmetrical, meaning if I rotated it 180 degrees and then said, hey, did I rotate this or not? You wouldn't be able to tell. So kind of the test is you close your eyes and I can either rotate or not rotate. And if you open up your eyes and it looks identical to you, then that's the type of rotation. So this first one, 180-degree rotation. The next one, it would be a 120-degree rotation. And each time I rotate 120 degrees, I come to an identical spot. And then they have some that are a little bit more complex. So this also has a rotational axis and a mirror image, I believe. Must be the end of the section. I'm losing my mind. But anyway, you've got an axis of symmetry and rotational symmetry there. Like I said, the whole thing that we just want to understand is that these proteins can orient in very complex things, but they tend to orient in a symmetrical arrangement. And one of the questions that they ask is, why can't it be mirror symmetry? So actually I said mirror symmetry. It's not mirror symmetry. We have axial and rotational symmetry. You don't find mirror symmetry in proteins. And that is a good question. I'm going to let you guys answer that. If you want to confirm your answer with me, you can, or it's in the book. It's a really interesting answer. And then you can get these higher ordered symmetries, such as tetrahedral, octahedral, icosahedral symmetry. Anyway, there's all sorts of these symmetry groups. And I just wanted to show you one of the proteins that I actually worked with. It's called alpha hemolysin. I put it on the first slide here. This is a homoheptamer. So it's this right here. It's a toxin released by Staphylococcus aureus. It is called hemolysin because it inserts in your membranes and causes the insides of your red blood cells to leak out and causes hemolysis, or lysis of red blood cells. And so there are 7 identical subunits that form into this mushroom-shaped pore. You get kind of this beta barrel here. And anyway, each one of these strands here corresponds to one subunit. And so those 7 subunits form into 14 strands that actually form this beta barrel down here. And this is very hydrophobic and sits in the membrane. And the cap sits up above the membrane. And if you looked at Oxford nanopore sequencing, this is what they sequence with. They actually run the DNA through here, and they look at the conductance of electricity through this pore. And essentially, as you send the nucleic acids through here, depending on the size of the base, you get different signals out. Anyway, this is what I spent my postdoc playing with, this protein right here. So kind of an interesting one. It's got this interesting quaternary structure, and it just looks really pretty. So we'll leave you with that, and the next lecture we'll pick up on section 6.4.